right. Well, thank you very much and for inviting me to do this presentation. Just essentially mostly to guide the discussion and also the great help of the AML Hub team to prepare the slides and get these things ready for you. So um, I think that we're all very excited about this new development in the treatment of AML. And it is very exciting to see the rapid development of these drugs. Some of them already in the market, at least in the U.S. and coming in other parts of the world. But also new drugs rapidly being developed, coming behind the ones that have already made it. So I don't need to tell you too much about the rationale for these. I do remember, however, that one of my very first manuscripts that I wrote when I was a fellow with my great mentor, Eli Esty, was about the poor prognosis of patients with 11q23 abnormalities. In those days, you were guiding it mostly by cytogenetics. And back in those days, which I'm pretty old, that was novel. That was a cool paper because it was starting to see why. And then, as you know, then it became known that it was this in those days called MLL1 gene rearrangement, and there had always been an attempt to try to develop an inhibitor of MLL1, which was not possible. However, with the development of the understanding on how these, what we call now KMT2A, works, that has become possible. And it is relevant to, at the moment, predominantly two mutations, although we know that there are others that may be where this same principle applies. NPM1, which is relevant because of its frequency, it's about a third of the patients, adult patients with AML, and KMT2A rearrangements because they're not as frequent, but they do have a very poor prognosis. And what happens is that KMT2A it binds to the DNA, or binds to menin, and through that it regulates the expression of the HOX and MEIS1, which promote the proliferation and leads to leukomogenesis. And then mutated NPM1 also binds to these interacts with these menin and KMT2A arrangement and results in the same leukomogenic process. So the menin inhibitors, by preventing or inhibiting this interaction between menin and KMT2A, they prevent this overexpression of HOX and then lead to normal hematopoiesis and differentiation, which is an important mechanistic understanding of how these drugs work, because they explain also some of the things that we are seeing in the clinic in terms of their activity and their some of the adverse events that we're going to be talking about in a few minutes. So there, as I mentioned, there have been several drugs that have been developed. The initial ones, the two that are leading the way are first revumenib. That was the first drug that was approved by the FDA. Initially, as monotherapy for refractory relapsed acute leukemia with KMT2A rearrangements. And shortly after that, the indication also as monotherapy for refractory relapsed leukemia for NPM1 mutated leukemias was added. Notably, this is approved for both adult and pediatric patients that are aged 1 or older. And then ziftomenib was, in between those two approvals, ziftomenib was approved by the FDA also as monotherapy, also for relapse refractory NPM1 mutated AML. Only that indication for ziftomenib. The KMT2A rearrangement is not approved for ziftomenib at the moment. None of these drugs have been yet approved by the EMA. although they have both been granted orphan designation and they're undergoing review. And I am sure in other countries the same thing is happening in different stages of approval. And this is, little by little going to become available for patients in clinical practice and not just in clinical trials. So I want to review some of the studies that are important to understand where we are and what these trials that led to the approval, and then follow that by review of upcoming studies with these drugs and also the data with the emerging drugs that we are starting to see. So let's start with revumenib. You also the initial paper that was published in Nature a couple of years ago, already showing very, very significant activity in these patients. So more recently, in Orlando last year at ASH, we saw an update of the Augment 101, which is the study of the monotherapy. And this update was specific for patients with refractory relapse KMT2A rearranged patients. As you recall, we had seen very good overall response rate. 2/3 of these patients responded. But the CR/CRh rate is relatively modest as a single agent, in 23% in this case. And the duration of remission is 7 months, a little over 7 months. Now, most of these CR/CRhs are 2/3 of them are MRD negative, so that is good. But again, the duration of response is good, and for this patient population, but certainly with room for improvement. In terms of safety, there's two main adverse events to notice with revumenib, and some of them apply to many of the other TKIs. Many of the other menin inhibitors. One is QTc prolongation in a little over 15% of the patients, grade 3. And the other one is differentiation syndrome, kind of the same incidence of grade 3. Now they have been manageable. I think that both of these events are things that we are more used to seeing with other drugs. We were talking about quizartinib, and certainly QTc prolongation is something we see with quizartinib. We were talking about IDH inhibitors, and that's something we became very aware of with the IDH inhibitors. So it has not resulted in treatment discontinuation, because I think that people are aware of recognizing and managing these adverse events. There's also this SAVE study, which was also presented at ASH. And this is for what was presented. This is a combination of revumenib with an oral regimen with the cytarabine, cedazuridine plus venetoclax combination. So a triplet in patients with newly diagnosed either NPM1 mutated or KMT2A rearranged AML. So these are small cohorts still. Overall 21 patients, 14 NPM1, 7 KMT2A rearranged. But the response rates are very attractive, 80%, around 80%, plus minus in both cohorts and overall. And the duration of remission, about 70 to 80%, are maintaining the remission at 12 months. All the CRs, all the responders are MRD negative, which is very attractive, and that includes even some of that are responding outside of the CR/CRh categories. And then again, we see they reported some differentiation syndrome, perhaps less incidence, and I think we've seen that with combinations, a little bit less of the differentiation syndrome, and no reports there on QTc prolongation. So very interesting results, both with the follow-up of the Augment and then of this triplet combination. So for ziftomenib, there was also some follow-up presented at ASH this last year. And this come from the COMET study, the COMET 007, which is the ziftomenib combination with Ven-Aza. And this includes cohorts with refractory relapse disease, as well as patients with newly diagnosed disease. What was presented for the refractory relapse is both the NPM1 and the KMT2R. There is a better response rate here for the NPM1. You see there with an overall response rate of 65%, and CRC, that includes CR, CRh, and CRi, 48%. Whereas for KMT2A, it looks like we've been seeing a little bit of a lower response rate with Zifto. Again, this is not approved. Zifto is not approved for KMT2A. So overall response rate 41, CRC of 28%. There has been no QTC prolongation, so much less of these with ziftomenib, and there's only been one grade 3 differentiation syndrome in this cohort. In the newly diagnosed, it's only NPM1 patients, and here the response rate is in 89%, the overall response rate, and the CRC is 86%. If you recall, I just showed you the equivalent combination with the revumenib, that one with the oral decitabine, and it, we're talking about 80s range in these kind of responses. Once again, just one instance of differentiation syndrome. This was a grade 2, one instance of QTC prolongation. So much less of that with this combination. There's probably perhaps less of with this drug anyway. Certainly less QTC prolongation, and less so with the combinations. So this is a summary of some of the studies, and of course this is rapidly changing, but these are some of the key studies that are being conducted right now with these two drugs. There's all sorts of combinations, mostly combinations with fludarabine and cytarabine, with Ven-Aza, with 3+7, and there's there's one study also with monotherapy, but interestingly that intercepts studies for patients in remission in CR1 through CR2 or CRi. With ziftomenib, we do have also the still ongoing patients. It's not accruing anymore, but it's still patients ongoing on the COMET-1, the monotherapy. But importantly, there is the combination studies. There's a refractory relapse study, which is called COMET-8, which includes arms with both intensive chemotherapy, FLAG-IDA. It's refractory relapse, so it's FLAG-IDA, or low-dose cytarabine. And there is one arm that has FLT3, that for FLT3 mutated patients includes also gilteritinib. So both gilteritinib and ziftomenib together with whatever chemotherapy is selected depending on the patient's characteristics. And then the COMET-17, which is going to be for newly diagnosed patients, both NPM1 and KMT2A. And again, it has a higher intensity and a lower intensity chemotherapy depending on the patient characteristics. These studies are enrolling, they're ongoing, and we'll be seeing data soon. Now, there are other inhibitors, as I mentioned, and this is a partial list. I highlight some of the ones that are more where we've seen a little bit more data, where we probably have seen more. But I'm sure you're all aware of and probably working with some of others that are coming along behind these. Bleximenib, we saw some data already in previous meetings, and there's not only the monotherapy study going, but very quickly going already into the combinations, 3+7 and Ven-Aza, and both in the refractory relapse and the newly diagnosed patients. Some of these studies are coming to an end, and we be seeing follow-up and results soon. Enzomenib, that was present at ASH, both in the monotherapy and combination with Ven/Aza for refractory relapse, both in NPM1 and KMT2A rearranged. The Balomenib, a little bit earlier, just as right now in healthy volunteers, and then BN-104, which is also already in monotherapy and combination studies, both for refractory relapse and newly diagnosed. So you can see that these drugs are quickly going, not only just through the monotherapy, but quickly going through the combinations and to the frontline therapy. So let's review a little bit of the data that's available with these drugs. So this is Bleximenib. Bleximenib, the trial is called the A102, and this was presented at EHA in June, and a meeting that unfortunately I missed thanks to the collaboration of Delta Airlines, but I'll leave that discussion for later. And then there was an update at ASH these past years. So on the left, you see here the combination with Ven/Aza, both for the refractory relapse and the newly diagnosed patients with AML, and for the two cohorts, KMT2A rearranged and NPM1. And as you can see, there's a very good response rate. Look at that overall response rate in the NPM1 refractory relapse. It goes to 90% plus. And if you only consider the CR/CRh, it's already 40%. And so it looks very, very good response rate. You cannot, it's very difficult. This is combination. So obviously we expect much, but it is a very attractive response rate. In the newly diagnosed, well, of course, we are also very high response rates in the 70s and 90s for KMT2A rearranged and NPM1. Now, granted, these are small cohorts still, so we need to see further follow-up and more data, but certainly this start looks very attractive. So far, there's no DLTs. There is very low rate of differentiation syndrome, again, combination, and no QTc prolongation grade 3. And then you have the combination with 3+7, and you see the same thing, both for KMT2A and NPM1, very high response rates in 90 to 100%. Again, small cohorts, but very attractive responses, very good safety with no differentiation syndrome, and just very little of QTc prolongation. Now, this is the Enzomenib that was presented at ASH this past year. This is monotherapy in refractory relapsed patients. And the first thing you see here is that the differentiation syndrome seems to be lower. It's just around 8%. There has been no dose-limiting toxicity, no grade 3 QTc prolongation. Very, very looks like a good safety profile. And then in terms of the efficacy, the overall response rate is the 70s, and the CR/CRh is 40% for the KMT2A. That looks very good. We'll have to see again. We're starting. There's 15 patients, but that CR/CRh is again, granted different studies, but looks very, very comparable to the other studies. And then you see on the right the NPM1, where again small numbers, but there's at least the survival and the remission duration. So this is a summary of all these studies that I showed you. And again, some of them are combinations, some of them are frontline, some of them are relapse. But I think we can say that certainly these drugs are valuable. They have modest activity, but very welcome activity as a single agent. But the combination strategies are looking very attractive nearly in all of these studies. And with differentiation syndrome and QTC being the main things to keep an eye on, looks like some of that is alleviated with the newer drugs and or with combinations with chemotherapy. So again, I think these results are very encouraging, a very welcome addition to the patients because it addresses for the NPM1 a significant number of patients, for the KMT2A a patient population that has a poor prognosis, and I think that the main question now is where do we go? How do we incorporate them into our therapy? And also how to expand it into other areas. I think the questions, for example, in the NPM1 frontline newly diagnosed patients becomes an interesting observation. How do you determine that those patients would benefit long term in the relapse refractory setting? Absolutely. Frontline, we will need a little bit of data to confirm the benefit over the combination. The toxicity, I think, is manageable, but I also think it is important to keep an eye on these because more and more these drugs are being used, certainly in the US, by our colleagues in the community, and the early recognition of differentiation syndrome is key to minimize the bad consequences. I can leave it there and let Charlie probably guide the discussion on these. But I think it'll be attractive to understand how you all see the incorporation in these patient population, and particularly for the frontline setting, because obviously that's where we want to go. We were talking about the same thing with the FLT3. We developed them in salvage, but you want to bring them frontline. And how do we do that, and what would it take to convince us to do these? And
okay, thank you
very much for your attention.
That was masterful. We've got a few minutes for discussion, so please, colleagues, please put up your hands or just shout out with questions. Whilst people are thinking, Jorge, I'd just be interested in, kind of the route to regulatory approval for these drugs, both in the fit and the unfit setting. And there are challenges in the NPM1 setting fit, aren't there? Because what's the control arm? And secondly, there is just this existential challenge about finding enough patients with KMT2A rearrangements, either fit or unfit. So I just wondered if you could make some comments about what trial designs should look like to generate the data that the regulators will need.
Yeah, for the NPM1, it unquestionably needs a randomized trial because they're not as scarce and you really want to see the benefit. It sounds attractive, sounds like it will work. It sounds like it can benefit patients. But these patients already, if it's just NPM1 and no FLT3 mutations, they do well. So we need to see that the benefit is real. And not only that the benefit is real, but, because I always see it in two perspectives. One is regulatory approval and the other one is use. That the benefit is real can give us regulatory approval. How much benefit there, what's the delta will also determine how much are we willing to work, because then there's added toxicity and cost and another drug, all these things that are part of that. But I think, that's a design of these pivotal trials. For the KMT2A, yeah, it's going to be a challenge with randomized trials because of the scarcity. I hope that regulatory authorities will allow, and it's probably a historical control is probably more reliable because the prognosis is poor, provided you have a significant benefit. But then the problem is if the patients are being transplanted, which largely they are, then how do you adjust for that if you don't have a proper control? So that is a question.
Thanks. And then just perhaps maybe we could bring in Brian Huntley, who has done a lot of work in this space, I think, just to talk a bit about resistance, mechanisms of resistance and how we might avoid them. Brian, did you want to make some comments?
Yeah, I suppose, I mean, I can do. Well, obviously there's genetic and non-genetic mechanisms that we can sort of invoke from the first trials. It sounds as if the from the experimental and from some clinical sort of anecdotal that there is potentially some sequencing that different mutations are specific to different inhibitors. And there's also some suggestion that some of the later inhibitors have an effect to degrade the protein, which in itself would be advantageous. With regard to the non-genetic, which seem to be the majority of the cases, there's evidence that other epigenetic regulators actually sort of take over re-establishment of the critical programs, particularly CAT6A and the PRC1 complex. So there are inhibitors of those complexes that are available. Most of the resistance has been seen in the monotherapy, so I think we have to be, cognizant about our hope that the
Yeah,
combined
exactly.
therapies will do much
Yeah.
better. But yeah, we have to be aware of resistance mechanisms and potentially plan for them. But hopefully the combination therapies will render those less necessary.
That's a great point, Brian. Oh, and Jeff, question.
Yeah, no, just to comment. Thank you, Jorge. That was a outstanding, comprehensive presentation. I don't know how you got so much information in 10 minutes. I wish I could mimic that. I just wanted to kind of re-emphasize, I think, the point that you made about the need to be mindful about how we design frontline studies, especially in favorable risk AML, where the bar is quite high, like the NPM1 population does extremely well with aza-ven doublet combination with high rates of MRD negativity, long survival. So really the question has to be, how do we best select patients for that type of a trial, keeping in mind the potential for resistance to develop, which could, abrogate the ability to successfully treat salvage in a salvage regimen with a menin inhibitor down the road, and knowing how well these patients do up front. So I think it's important to really be very careful about how we design these phase 1 trials, and maybe, consider, different risk categories within the NPM1 group that might benefit most from, an upfront treatment strategy with a triplet compared to maybe a more favorable NPM1 group that may not need it right away and might actually potentially have a more of a negative effect down the road with treatment options. Thanks,
Jim. That's very important. So we're slightly up against time, but I have one last question for Jorge, for which Jorge there is no answer. But if you had a 40-year-old fit young guy with KMT2A rearranged AML presents to your unit now, you're going to want to take him to transplant, I think. What's your, and you can have any drug you want, what's your preferred approach to getting him to transplant?
You said a 40-year-old?
Yeah, 40-year-old fit.
Yeah. Well, right now I have the trials open, so I would definitely put him on a trial with a 3+7 plus a menin inhibitor, but definitely proceed to transplant once I have them in remission. We've seen the remission rate is very high, and a lot of these are MRD negative, so that's the perfect scenario for these drugs, I think. So that's what I would do.
That's 3+7 with menin inhibitor. What about giving Ven/Aza with menin inhibitor?
Well, we don't have data for those patients because we haven't done it. I am a firm believer that that's where we're going to be heading.
Yeah.
But right now, I think the most conventional approach would be 3+7.
Yeah, rather unfashionably, you'd like to see a bit of data, right?
Yeah.
Okay, that was a really fantastic talk. Great discussion. Thank you again, Jorge. Brilliant stuff.